Hi everyone, very good evening all of you. Hello, good evening all of you. Hi, this is Vaishali Kamdar and today we will be going to discuss about biomolecules. So let me know, can I have some quick comments please if you can hear and if you can see the video clearly? Yes, those who have joined, quickly join faster, and those who have joined, type quick message in comment section, in chat section, that you yes ma'am, you are audible and your video is visible. Yes guys, come on, quick comments please. Am I audible clearly? So hi, this is Vaishali Kamdar and I'm from Spark Academy. So as you guys are following Spark Academy on sparkacademyonline.com and Facebook, Instagram, Telegram, wherever you would like to join, you can follow us and follow us on any of the platform, whatever platform is convenient and convenient for you for tips and tricks. So this is our free live classes on daily basis. We are conducting free live classes for MSET J and Neet. For MSET J and Neet on daily basis. So I'll be sharing you schedule. I'll be sharing the schedule with you. So what is the schedule and when you will be having chemistry, physics, maths and bio classes. All these classes schedule will be shared to you. Or else you can do one thing, see, download our app, and if you are still worried about your NEET exam or whatever MSET exam, so you can right now you can go for admissions, classroom coaching as well as online coaching. So short term and regular courses are already been they are running since long, but crash course we have started, offline crash course we have recently started, that is last week on 18th April. It has been started on 18th April. Still it's going introduction part. So if you have still not joined, then you can join the crash course, offline crash course, and you can join online coaching also. Online shortly and soon coming online coaching will be starting. Online admissions for crash course will be starting soon. So dates will be updated soon. So for that you need to contact the number scrolling in your screen. Number scrolling over your screen. Okay guys. So this is or else you can download the app, that is Spark Academy app. This app, and then you will be having access to whatever courses are going on right now, what will be the next, so you can select from that also. So this is our schedule, Monday to Friday, daily 3 hours, 5 to 7 evening time, and 11 to 12 morning. So 11 to 12 morning, 5 to 7 evening. So this is the schedule. So you can just take a screenshot and you should not miss out any of the free live sessions. Or else what you can see, one thing you are already knowing, that you can go to YouTube and at Spark you can subscribe to our channel, that is Spark Academy channel. And you can press the bell icon for the notification. So you will get notification on your phone. Otherwise, whatever classes you don't want to miss, you just connect to this schedule. You keep this schedule with you, and you can directly solve your doubts in chat section. I'm here to solve all your doubts. So this is my coupon code if you want to enroll for online or offline coaching. So this is my coupon code, that is VAI10. So this code you can apply and you will be getting great discount over it. See, this is my code. Again I am writing. It's VAI1010. So you will be getting discount on whatever fees. So, and you will be connected directly to Vaishali. So that's me. And you will be listed, you will be enlisted in my student list. So for today we will be going to discuss about cell and cell cycles. You all know this is short and sweet, very sweet unit. And we have discussed about the cell also last week. So this unit we have started last week. The cell we have discussed 2 days. And as it is very short, sweet, and you are well aware about all the type of topics already from 8th, 9th, 10th standard. So this is what we will be going to discuss here. The cell has been completed. Now it's about biomolecule, and next lecture it is about cell cycle and cell division. So altogether it will give you short three lessons only, and it will give 6 questions. That is not very less. 6 questions, 6 fours are. So 24 marks. It is good amount of the marks you will be getting with 24 marks if you are solving this unit. So from cell structure and function, the cell has been completed. Now it's about biomolecule, and from that you might expect 2 questions from biomolecules. Okay guys, biomolecules are there. This chapter is there in chemistry also, but in that you will think it is like in the as chemical point of view, chemistry point of view. Here we'll be discussing at biological point of view. So these are biochemist. This is the chapter of biochemistry. So that's why it is included in biology as well as chemistry. So it's there. So that's why this chapter is very important, because you will be getting questions in chemistry also from this chapter. And somehow they might be related, because same biomolecules, we don't have chemical or chemistry biomolecules differently, separately, and biological biomolecules separately. So it's all together, it's biomolecules only. That's why it's very important, because you will be getting questions in chemistry as well as in biology also. So biomolecules. Biomolecules are divided into 2 main types based on their molecular weight: biomacromolecules and biomicromolecules. Macromolecules, name itself suggests, these are macro, that means big, and small, that means micro. So proteins, polysaccharide, nucleic acid, they are included in biomacromolecule. Or we can say one thing, that means biomolecules which are having molecular size greater than 1000 Dalton, then they are part of biomacromolecule. If it is less than 1000 Dalton, then it is known as biomicromolecules. Okay, those size of lipid may be smaller as compared to other macromolecules, but lipids are included. See, lipids are part of acid insoluble fraction. So now what is this acid soluble insoluble fraction? How you will get idea about all these molecules present in our body or in any living tissue. So let me discuss with you one experiment. Wait a second. So first, how you will identify which biomolecule is there in a living tissue. So take any living tissue, whether it is plant, animal, any tissue. So tissue, it is along with tissue is grounded. I mean, it is grinded along with trichloroacetic acid. Trichloroacetic acid. With this acetic acid, it is grinded by mortar and pestle. Okay, simply chutney banate hai वैसे. So like that only you are making chutney of tissue using trichloroacetic acid. Will be not using any other juices or anything else. So we will be using acid here and you will get slurry after that. Now what happens to the slurry? Now tissue plus trichloroacetic acid gives you slurry by mixing. So slurry, it is now you are straining the slurry in muslin cloth, cheesecloth, muslin cloth or cotton cloth. So you will be getting simply when you are straining chai, tea. At that time you will be getting tea in your cup and the chai patti in your strainer, right? So like this chai patti, why you are not getting inside your chai ka cup? Because patti, it is having molecular size of patti, it is larger than the pore size of the strainer, right? So it will be there in retentant part, and so that you will be getting two type of fraction here. Two fraction you will be getting. One is called retentant and one is called filtrate. Filtrate, that means simply your chai. Okay. Filtrate, that means whatever is strained, or you can say it is acid soluble fraction. Whatever molecules are soluble in acid, simply when they become soluble, they will be strained from the cheesecloth. So you will be getting them in form of filtrate, that is below that. And in your strainer, over your strainer, whatever you are getting is retentant part, that is containing acid insoluble fraction, that is also known as acid insoluble fraction. Okay, I hope you are understanding, guys. If you are not getting this point, let me tell me if you are not getting this point. I will explain it again. Okay, simply just like straining anything. Okay, when you are making juices, like juice, watermelon juice. Right now it's summer season, we used to have watermelon juice. So just simply grind watermelon in mixer, but here we will be not using electric mixing. Okay, electric mixer grinder, we used to do it with mortar and pestle, that is just like how we make chutney. Okay, so we should not use water over here. We want one liquid to mix it, to grind it properly. So for that we are using trichloroacetic acid. Remember, this is trichloroacetic acid. So after that you will get a slurry. Slurry is strained in cheesecloth. That's how you will be getting two fractions. One is retentant, another is filtrate. Filtrate is here. For example, this is your beaker, and in that you will be getting filtrate over here. This is your strainer. So you will be getting another fraction, that is higher molecular weight fraction or higher molecular size fraction here as retentant part, as it has been not soluble. It is not soluble in acid also. So that's why we are getting two fractions, acid insoluble and acid soluble. Okay. So we have one exception over here. Lipid being low molecular size, still it will be found in insoluble fraction. Still it will be there in form of insoluble fraction. Okay. Why this lipid is having low molecular size? Because lipid is part of cell membrane. Okay, lipids are part of cell membrane, and when you grind it, during that cell membrane has been ruptured, so that's why they are converted into smaller pieces. So that's why they are having small size, but still they'll be found in insoluble fraction. Okay, otherwise retentant will be containing macromolecules. Macromolecules, that means polymers. That may be inulin, lysine. So inulin, so lysine and lysine, it is amino acid, fructose, it is sugar. So that you will not get there. Okay. Inulin, it's a polymer. So these type of polymers you will get here in form of acid insoluble fraction. Then what is there in acid soluble fraction? That is biomicromolecules like amino acid, monosaccharides and nucleotides. So these are micromolecules, simply micromolecules. They are, they ask you to do like this. Hold your ear with a twisted manner. What is micromolecule? Micro itself suggests it is small molecule. Small, that means monomer part of a polymer. It is smaller. Simple. Monomer part of polymer is small only, na? So proteins are made up of amino acid. Polysaccharides are made up of monosaccharides. Nucleic acids are made up of nucleotides. So all these monomers, they are called biomicromolecules. Okay. So I hope this much is clear, guys. So now, see, we have another type of, we can say, acid soluble, acid insoluble, that is true. Along with that, this tissue might contain certain inorganic compounds also. Inorganic compounds also. See, this is about organic one. This protein, nucleic acid, all of these are organic. Now, what about inorganic one? See, for that we will be using destructive method to identify which inorganic material or which inorganic element it is there. Okay. Which method? Destructive method. So in this destructive method, simply you are weighing the tissue, wet weight, that is known as wet weight. Now you will allow it for drying process. So dry it, that will give you dry weight. Now this dried tissue, it is burned. So after burning, all the carbon dioxide, all the organic molecule will be evaporated in form of carbon dioxide and water. Whatever you are getting in form of ash, that is your inorganic compound. That is showing inorganic substance present in your living tissue. So this is very important as Neet point of view. Very, very, very important, guys. So very important. Wait a second. I want to highlight it. So very important for Neet point of view. Important for Neet point of view. Ash gives you idea about inorganic substance present in your living tissue. And how you will get idea about organic substance? For that you need to make slurry in trichloroacetic acid. Then you will get two type of fraction, acid soluble and insoluble. I hope this much is clear, guys. Now we are discussing biomicromolecule. In that first it is about amino acids. So what are amino acids? Amino acids, they are actually, you can say, substituted methane. What is methane? CH, H, H and H. CH4, it is methane. Okay, now here one hydrogen is substituted by carboxylic acid. One hydrogen is kept there only. Another hydrogen is substituted by R group, that is variable group, side chain. And this is substituted by NH2 group, that is amino group. So substituted, what are amino acids? They are actually substituted methane. Okay. So this amino acids, they are of three types: acidic, basic and neutral amino acids. So on the basis of how many molecules, so if they have higher to more than one COOH, more than one carboxylic group, then acidic, more than one NH2 group, then basic, both are equal, present in equal number, then neutral. Of course, you might get idea from this. See, amino acids have this Zwitter ionic form also. What is Zwitter ionic form? See, you can see here. So there is one more hydrogen added here. This is positive ion. Okay. Here it is negative ion. You can see from here. So this is negative. Okay. And this is intermediate compound where positive, it's a positive also and negative also. Both the charges are there. So that is known as Zwitter ionic form of amino acid. So at whatever pH, what is Pi? Pi stands for pH at which you are getting Zwitter ionic compound, Zwitter ionic form. You get Zwitter ionic form. That is called Pi, isoelectric point. Point at which you are getting Zwitter ionic compound, Zwitter ionic form, that is known as Pi. Okay, so amino acids are a type of micromolecule, biomicromolecule. Then second it is acids. Second is lipids. So lipids are esters of fatty acids with alcohol. So fatty acids with alcohol. So they are insoluble in water and soluble in number of non-polar organic solvents like benzene, chloroform, ether, etc. Lipids form colloidal complex and get dispersed uniformly in water in the form of minute droplets. The complex is known as emulsion. When you mix oil and water and you stir, keep on stirring it, still you will find minute droplets of lipids. So that is known as emulsion. Okay, so lipids equal to fat plus alcohol. What are lipids? Is equal to fats plus alcohol. So basic component of all the lipids are fatty acids. Now functions of functions of this lipid. Fat stored in adipose tissue serves as the efficient source of energy. Whenever you are on fast, right now Ramzan is going on. So when you are on Roja, at the time, Roja ke time pe, at the time, from where you are getting energy? Yeah, that's true. After sunset, you will be having food. That's true. But see, in some religion, they are doing fast continuously. So they'll not eat anything during the day or night also. So that is complete. They are on fast for 1 week, for 20 days, for 1 month. So how they are getting energy? So at that time, whatever tissue, adipose tissue, they have stored fat, they have stored energy in form of fat. These fat is getting dissolved and from that you are getting energy. This fat will go for respiration and from that you are getting higher amount of energy. Okay, though it's required lot of it's required time to dissolve this fat. So fat serves as a thermal insulator and subcutaneous tissue around the internal organ. See, those who are having high layer of fat below their skin, that means subcutaneous tissue, they will not, during cool environment also they will not feel cool. Okay, because of this thermal insulator type of, it is serving as a thermal insulator. So they also act as an electrical insulator against nerve impulse transmission. And cholesterol and phospholipids are important constituents of cell wall. See, what are lipids actually? Glycerol. What is glycerol actually? Glycerol is, when you think about glycerol, it is actually trihydroxymethane. That means like this. Okay, it is trihydroxymethane. 3 hydroxy groups are there. So when lipids are formed, what are phospholipids and what are lipids? So at that time this glycerol is esterified with fatty acid. Fatty acid can be different on the number of side chain it is containing. So this esterification, that means this hydrogen is removed and there one RCOOH has been introduced. So it is binded like this. This is ester bond. O, this bond is known as ester. So this is how esterified glycerol has been esterified by fatty acids. And if esterification, during this esterification, phosphoric acid, that is PO4 or PO3, it is also attached there, then it is forming phospholipids. This phospholipid, they are main component, main constituents of the cell membrane. One of the well-known phospholipid is lecithin. Now next it is sugars. So sugars are sweet and water-soluble carbohydrate. So sugars are water-soluble carbohydrate. They are formed by CHO, carbon, hydrogen and oxygen, in ratio of 1:2:1. So you can see two type of sugar here. One is pentose, one is glucose, that is hexose. So 5 carbon compound and 6 carbon compound. So there are two types of sugars also, reducing and a non-reducing sugar on the basis of the free aldehyde and ketone group if it is present. See, let me reduce. So two types of sugar, reducing and non-reducing. So reducing sugar, that means contains free aldehyde or ketone group, free CHO or COO group. So that is known as reducing sugar, those who are not having free aldehyde and ketone group. Not having free aldehyde and ketone at the end, then they are called non-reducing sugars. Then nitrogenous bases. Nitrogenous bases, they are carbon compound with heterocyclic ring. So adenine, thymine, guanine, cytosine. So it includes purines and pyrimidines. We have purine, purines, adenine and guanine, pyrimidines, cytosine, thymine and uracil. So see, I can give you one. See, this is trick for today. This is a tip or trick to remember nitrogenous base. So that is cut COT, cut pyrated because silver is pure here. That means cut, that is CUT, cytosine, uracil and thymine. This cytosine, uracil and thymine. These three are pyrated, that means pyrimidines. And silver, what is symbol of silver in chemistry? Ag. So that is adenine and guanine. They are pure, that means purine. Okay, adenine and guanine, they are pure, that means purine. So you can remember from this structure also where exactly nitrogen is present in pyrimidine, third and first position. Okay, so you can think the double ring structure having fused ring. In purine and pyrimidine, it is single heterocyclic ring. Then nucleoside and nucleotides. So nucleosides, nitrogenous base plus sugar gives you nucleoside depending on the different nitrogen base. So there are different nucleosides. Adenosine, guanosine, cytidines. Question can be asked. Most important point to note here. Question can be asked here on the basis of these nucleoside and nucleotide. Important. And this is also important. In question, they might give you that guanylate acid. It is nucleoside or nucleotide of what? So in option, they might give you it is nucleotide of this or nucleoside of this. So that means you need to select it as nucleotide. Guanosine is a nucleoside. So remember names of nucleoside. Ine when it is suffix Einstein, guanosine, cytidine, thymidine, or it is written. So then it is known as side, nucleoside. See, for nucleoside we have ion. Okay, and for nucleotide we have acids. When acid is there, it is nucleotide. I hope this is one more trick for today. So adenosine, uridine, so like that, these are nucleosides. When this nucleoside, they are attached with one phosphate, that is forming a nucleotide. These nucleotides, they are actually monomer of nucleic acid, that is DNA, deoxyribonucleic acid, and RNA, that is ribonucleic acid, depending on the sugar present there. So if sugar is not having one oxygen at second position, that is forming deoxyribose sugar. Then macromolecule. So in that, first it is protein. So proteins are higher molecular weight polymers. Proteins are very variously folded linear heteropolymer of amino acid. We will discuss during genetics that transcription and translation. When translation occur, after translation, you will be getting polypeptide chain. See, this simple linear polymer of amino acid. Simple linear polymer of amino acid. Linear polymer of an amino acid gives you polypeptide. This is not a functional protein. Okay, this needs to undergo certain type of modification, certain type of folding. So that will give you functional protein. So protein having two or more polypeptide is called multimeric protein. Two or more polypeptide, that is known as multimeric protein. Peptide bond is a covalent bond formed when COOH group of one amino acid reacts with the amino group of the another amino acid. See, what is the structure of amino acid? Let me draw here. See, like this, it is substituted methane. So COOH, here it is NH2, H and R, that is variable group, plus again NH2, COOH, H and R. So here like this, one OH and one hydrogen is removed like this, so that will remove one molecule of water. And which bond is formed? You can see CONH bond is formed. This is called peptide bond. It's a type of covalent bond. It's a type of covalent bond which is known as peptide bond. From COOH, one OH group is removed, and from NH2, one hydrogen is removed. So HOH together form H2O. So by releasing, so that is known as dehydration. So group next amino acid releasing one molecule of water, which is known as dehydration. Then this polypeptide chain will undergo primary structure formation. So linear sequence, that is primary structure. So linear sequence of amino acid in a polypeptide chain. It describes the sequence of amino acids, the positional information in the protein. The left of the chain has first amino acid. So that is known as N-terminal amino acid. You can see here. This is, for example, this is first. For example, this is first. Then you can see here nitrogen is there. So this is called left side, it is called N-terminal, and right side it is called C-terminal end. Okay. So left end of the chain has first amino acid, that is also known as N-terminal amino acid, and right hand side of the last amino acid, it is also known as C-terminal amino acid. So which groups will be free? Which will be the free groups here? So that this is having NH2 group on this side. At the end, and this is having COOH group, that is carboxylic group at one side. Then it will undergo secondary structure formation. So secondary structure includes beta pleated sheet and alpha helix formation. So you can see here alpha helix and beta pleated sheets. So and collagen helix also. So in alpha helix polypeptide chain is collides spirally. So spirally helix-like structure is formed in right-handed manner. Remember, alpha helix is right-handed manner. So the helix, that means when you start coiling it, it is towards the right-hand side, not towards the left-hand side. Okay, so helix is stabilized by hydrogen bond between two amino acids. So keratin, myosin, epidermis, fibrin, etc. They are alpha helix form of secondary structure of protein. Then beta pleated sheet, that is also secondary structure. Two or more polypeptide, they are interconnected by hydrogen bond. So it's simple, interconnected, they will be forming sheet-like structure. A sheet is produced instead of fiber or a rod or a helix. So beta keratin again here. So it is beta pleated sheet, that's why we are using beta keratin, fibroin and silk. Remember, fibrin is alpha helix and fibroin, it is beta pleated sheet form. Then collagen helix, three strands of the polypeptide chains are coiled around one another, So that is three strands. Batch of photos. At that time you require more than three photos at least. Okay, that will give you collage. So remember, like that only, this is the tip for today. So that is collagen helix, that means more than three strands together forming they are coiling together, gives you secondary structure, that is collagen helix. Then tertiary structure. Hello guys, I hope I am audible. So tertiary structure, the helical polypeptide chain is further coiled and folded to form a complex structure. So that will give you tertiary structure and gives 3D view. This is tertiary structure. This is secondary structure, and this is tertiary structure. Okay, so it gives you 3D view of protein. And tertiary structure is necessary for the many biological activities of protein. Various types of bonds are found during the coiling of the polypeptide, and they are covalent bond, ionic bond, electrostatic bond, hydrogen van der Waals interaction, and hydrophobic bond. So any of these weak bonds are involved during formation of tertiary structure. Then last, that is formation of quaternary structure. Quaternary structure, so like hemoglobin. So that means when heme component it is there, around the iron 4 globine chain it is attached. So that will give you quaternary structure. Still it will folded more. Still it will be going under folding, more folding. And it is combined with one or more polypeptide chain, more than 3 polypeptide chain. They are attached together and they are forming quaternary structure of it. The next macromolecule, polysaccharides. So polysaccharides are condensation of polymers. Polypeptides are condensation of polymers in which monosaccharides are held together. Through glycosidic linkage. Remember the linkage here, this is not peptide bond, this is glycosidic linkage. So they are colorless and tasteless, and so they are called non-sugars. They are called non-sugars. They are concerned with the two important functions, structural and storage of the energy. So two important functions here, storage of energy, and structural component also They are also structural components. So polysaccharides, remember they are tasteless. Though monomers are sugar, they are sweet. But this sweet together linked by glycosidic linkage, then they'll give colorless and tasteless polysaccharides. Glycosidic bond in polysaccharides. It is the bond formed when individual monosaccharides are linked between two carbon atoms by dehydration. It is formed normally between carbon atom. It is formed normally between the carbon atoms 1 and 4 of neighboring monosaccharides. So that's why it is also known as 1,4 glycosidic linkage. 1,4 glycosidic linkage. So then nucleic acid. Nucleic acids are polymer of nucleotides as we have discussed. So nitrogenous bases are of two types. Purine pyrimidines are there. Sugars also are of two types. So two types of sugar, two types of nitrogenous base, purine pyrimidine, sugar types, ribose and deoxyribose, forming RNA and DNA respectively. So phosphate molecule links the third carbon atom and fifth carbon of the sugar. This linkage, it is known as phosphodiester bond. So phosphodiester bond is present in DNA as well as RNA. So it's a strong covalent bond between phosphate and two sugar groups. There are two types of nucleic acid, DNA and RNA. Next, enzymes. So enzymes are of two types, simple, made up of protein, and conjugated. That means they have protein part that is forming apoenzyme, but they require some cofactor also, that is non-protein part. This together form a conjugated enzyme. Okay, so enzyme, see simply how enzymes are working. See, enzymes, they will form, see for example, this is one enzyme. So it is having one active site here. This is its active site here. So here one substrate molecule will bind. So substrate is specific for that enzyme only. Okay, so it will bind like this to form EX complex. That is, this is how it will be. Okay, this is how it will be. When ES complex is formed, there is slightly modification in the structure of enzyme occurs. Because of that, it will tightly fix to the substrate. So this is enzyme, this is substrate, this is ES complex, and it will in turn gives you product and free enzyme. First EP complex, then it will separate in form of enzyme plus product. Okay, so what are enzymes? They are called biocatalysts. That means in presence of enzymes, rate of particular reaction increases. See, for example, carbonic acid. It is made from carbon dioxide and water. Simply soda जो बनाते हैं बाहर बंडी में. So like that, and that water and carbon dioxide are added without, they are forming carbonic acid without using an enzyme, without using any enzyme. So in that case, they might form, so without enzyme, it will give you around 2000 molecules of carbonic acid per hour. But with enzyme, it will give you around 6 lakhs molecules of carbonic acid per second. So this is how enzyme or carbonic anhydrase, it's in presence of carbonic anhydrase, this activity of or this reaction, it is enhanced, the rate of reaction has been enhanced. So this is the function of catalyst, or catalyst found in a biological body, it is known as biocatalyst. Okay, so in that case, when ES complex is formed, activation energy for formation of product that require it is less. So this is ES complex transition state formation. Okay, so acting without enzyme activation energy you can require, that is this much activation energy you will require without enzyme. But with enzyme you will require only this much of energy with enzyme. Okay. This enzyme activity, so enzyme activity can be depending on the different factors like temperature, pH, substrate concentration and enzyme concentration. Simply temperature increase, enzyme will be getting denatured. Okay, and if it is very less temperature, then it is in its inactive state. pH, it is similar to that. At higher pH, it gets at lower pH, it gets denatured, as well as over the higher pH also. Substrate concentration, when you increase substrate concentration up to certain level, the rate of reaction increases. So, up to certain level, rate of reaction increases because substrate is there, enzyme is also there, they'll bind completely. But after some time, there is no more enzyme available to bind with substrate concentration. Then it will go under steady state. Same happens with the enzyme concentration also. If substrate is not there, then it will go under steady state. So classification and nomenclature of enzyme. So there are different types of enzyme, that is oxidoreductases or dehydrogenases. That means they are involved. What is their function? Their function is oxidation and reduction. Oxidation and reduction. Then transfer is transfer of one group from one place to the another, from one molecule to the another, that is transferase. Hydrolysis, they break down a molecule with H2. So breaking a molecule by using water molecule, that is hydrolysis. Lysis without H2. They also break it down, but it's without H2. Then isomerases. Isomer, simply they are converting it into isomer form. Okay, ligases, they are doing gluing activity, molecular glue. So let me remind you, all of you, download our app, that is SPARKACADEMY app, and you will get discounts on all our courses. For that you need to apply this code again and again. I'll say that is VAI. To get 10% discount, to get discounts on any of the course. This is how after that you will be getting free handwritten notes, study materials, PYQs and free mock tests available. So link is there in description box. You can check the description box. So full length, all these things are available in description box. You can purchase, you can buy any of the thing from it. Okay. Courses, if you want to enroll for the courses, or else you can go for the PYQ solution or full length test or study materials also. So we are open right now for classroom as well as online coaching. Classroom coaching is going on, short term, regular, both the batches are going on, and this is not coming soon. Now it has been started, only till the introduction part it has been done. So you can join. It is not too late to join this course, crash course. So it has been it has been started from 18th April, that is last Monday. 18th April, that is last Monday. So you can join right now. Do not delay now. So just download the app, apply the coupon code to get discount, and enroll right now. This is not too late. Now you will do. Now you will wait for some time. Then it will be late. Okay. Because Neet dates are already has been declared. All the dates has been declared. So now you can join for the crash course. Now you do not delay now. We have two branches, Narayangura and Mehdipatnam. So whatever is convenient for classroom coaching, you can go for that. Online coaching also. For that also you can apply the same code. For that also you can apply the same code. that is VAI 10, that is 10. On daily basis, we'll be having the live sessions. All the sessions are in recorded. I mean, that is not recorded. We are doing live sessions, and that we will keep it in recorded form also. So you'll those sessions, those lectures will be available afterwards. So you can see right now on your screen. See how these lectures are available date wise. So whatever lectures you are missing, you or one or the other reason you have missed that session, you can check in your app also. So this is how we are giving assignments on daily basis. We are conducting test also, and test analysis also has been done. This is our method. And why Spark only? So that idea you might have got that idea from our YouTube classes. So we have highly experienced faculty and personalized coaching is there. Process oriented coaching on daily basis. We are pushing our kids. We are pushing our students to reach their targets. And we have limited batch size. This is a feature. If you opt for hybrid coaching, that means offline as well as online, you can have access to both the things. But for online, you could not have access to classroom. And you will be not having books in that case. You will be having online materials. Now, first question for today. Phosphoglycerate is always made up of what? Phosphoglycerate is made up of what? It's a type of lipid. It is made up of what? Phosphoglycerate. See, simply name itself suggests phosphate should be there, glycerol should be there. But how it will be? See, glycerol, I have told you it is trihydroxypropane. That means propane, that means 3 carbon. And trihydroxy, that means CH2OH, CHOH, CH2OH again. So this hydrogen has been esterified first with phosphate. Okay, then with fatty acid. Okay, so that means a saturated or unsaturated. Saturated, that means having single bond only. Unsaturated, it might contain double bond. Saturated or unsaturated fatty acid esterified to the glycerol molecule, to which one phosphate group is also attached, that is giving you phosphoglycerate, a type of phospholipid. Then second question, which of the following statements is not true for the cell membrane? See, remember, always read the question twice, so you will get idea that they are asking not true. Not true, that means you need to select wrong one here. So it is present in both plant and animal cell. Yes, it's true. Lipids are present in bilayer. Correct. Proteins may be peripheral or integral. That is also correct. Then carbohydrates are never found in it. This is wrong. So this is our correct option. Carbohydrate glycosylation also there in case of glycosylation. It is also there in case of membrane. Okay. In cell membrane structure, carbohydrates also present. Correct, Bhavna. Next, select the option which is not correct with respect to enzyme action. So again, not. So focus on the word not. So wrong one you need to select. So these are PYQs. These are PYQ already it has been asked in 2014. So with respect to enzyme, which one of these is not correct? Come on, Bhavna. Substrate binds with the enzyme at its active site. That is true. Addition of lot of succinate does not reverse the inhibition of succinate dehydrogenase by malonate. See what happens here? It is competitive inhibition. It is competitive inhibition occurs with, correct, Bhavna. Competitive, it's a type of competitive inhibition. So if you add more succinate, that is substrate. So more substrate is available, then malonate cannot bind. So it will reverse. See, this not is not correct. That's why we need to select this option. Next, macromolecule chitin is phosphorus containing polysaccharide, sulfur containing polysaccharide, simple polysaccharide, or nitrogen containing polysaccharide. No, it's not simple polysaccharide. That's not right. Macromolecule chitin, it is nitrogen containing polysaccharide. Chitin is made up of N-acetyl glucosamine. So glucosamine, amine itself suggests it has nitrogen. So N-acetyl glucosamine is there in chitin. So that is nitrogen containing polysaccharide. Then next, transition state structure of substrate formed during an enzymatic reaction is transition state. I'm waiting for your answer, all of you. It is transient and unstable. It's a transient and unstable state. It is not stable, that's why it will be releasing product and enzyme. Essential chemical component of many coenzymes are essential chemical component of many coenzymes. That means cofactor of many coenzymes. So they are vitamins in case of NAD and NADP. It is niacin, which is vitamin, which is acting as an essential component of coenzyme. Next, which of the following biomolecule does have phosphodiester bond? Very simple, easy peasy types. Come on. Yes, very simple. Nucleic acid and nucleotides will be having phosphodiester bond in it. Then which one of the following statement is incorrect? Which is incorrect here? Incorrect statement here, it is presence of competitive inhibitors. Competitive inhibitor decreases the Km of the enzyme for the substrate. That is incorrect. Then next one, which one of the following is non-reducing carbohydrate? That means not having free aldehyde or ketone group. Sucrose is a type of non-reducing carbohydrate. Now again, let me remind you, all of you, that you can join, you can follow us on Telegram link. Also, you can click on this link and join us on Telegram. First question, first homework question. Take a screenshot and I'll be waiting for your answers in comment section. So don't forget to send comments, okay, Bhavna? Plastids. Plastids differ from mitochondria on the basis of which of the following feature? Then next, enzymes, vitamins and hormones can be classified into single category of biological chemicals because all of these are Enhanced oxidative metabolism are conjugated protein are exclusively synthesized in body of a living organism. Take a screenshot, select your answer and write 1 ABCD, 2 ABCD like that. Last question, which element is normally absent in protein? In proteins, which one of these is absent? Carbon, nitrogen, sulfur or phosphorus? Select your option and answer. Subscribe. Don't forget to subscribe to our channel and press the bell icon for the notification. Don't forget to like, share and subscribe, guys. There is one thumb. So do not forget to press that thumb, that is like, if you are liking this video. So this is our MSET result last year, Neet result of last year. Thank you so much. Don't forget to like, share and subscribe.